Broccoli. It's about the healthiest vegetable you can eat. The only problem is it has a very short shelf life. But now scientists at Warwick's Horticultural Research Institute are trying to do something about it.
The problem with broccoli is that when it's harvested, it doesn't store for very long on the supermarket shelf, and then when you buy it and you take it home, it doesn't store particularly long, even when you put it in the fridge. So here, for example, we have 6 different lines of broccoli that are part of our breeding population. They're all harvested on the same day, but as you can see, they show very different time to yellowing. Shown on this graph here are 50 different members of the mapping population, and each of these lines is a different member of the population. This is the good parent, so this is the parent that stores really well, and this is the bad parent that stores really badly. In a broccoli plant, there will be at least 30,000 genes, many of which will be being expressed at the same time. On this slide, we've got 15,000 broccoli genes that we know are being expressed in broccoli, and we can use this slide to find out how those genes are being expressed and compare expression with the good storage broccoli line and the bad storage broccoli line. On the screen here is shown the slide, and the colours illustrate the expression levels of the individual genes in the different broccoli lines. So if I zoom in on this slide, you can see each of these spots here represents a different gene, and the colour of the spot illustrates the expression level of that gene. So a gene that's expressed really strongly in the good broccoli will look red, a gene that's expressed stronger in the bad broccoli will look green, and genes that are expressed at pretty much the same level in both broccoli lines will look yellow. The reason we've been funded for this work is to try and develop broccoli that will store for longer. This would be an advantage to the supermarkets because it would mean they wouldn't waste so much broccoli, they wouldn't have to throw broccoli away. It would also be an advantage to the consumers because the broccoli would store longer and hopefully remain nutritious and taste better.
Well, the genetic analysis goes on here in the Genome Centre at Warwick HRI. On this machine here, it's operated by geneticist Jason Pohl. Jason, how does it work?
Right, basically what happens is that you have a mapping population consisting of individuals that came from a cross between the 2 parents, one that has the trait you're interested in and the other one that you'd like the trait to be in, for example. And what we do is we take that population and we chop the DNA up using enzymes, and then we attach a label that allows us to visualize these chopped up bits of DNA, which we call fragments. And we run these fragments through this machine here, and it contains a dye, a blue dye, that can be visualized and recognized by this machine. Then what we get is a trace data files, which I can load up on my PC, and I can show you them here now. We have the 2 parental lines. For example, let's say this one has a long shelf life and this one doesn't have a very long shelf life. So they're the parents of our cross, and you can see that we have a peak here where the machine recognized a fragment. And if we look in the population, we can see that the fragment does not exist in this individual here, but it is present in this individual here, and it isn't here. And that's what we call a polymorphism, or a marker. And what we can do with those markers is we insert it into another program that looks at the frequency that these markers are inherited together. And what it says is each one of these represents a marker, and it says if this one is always inherited with that one in the population, then they must belong together on these units we call linkage groups. If they're not inherited together and it's completely random, it says that these markers must be present on another linkage group. And then what we can do is we can look at the phenotype data of the mapping population, and that's how long is the shelf life of that particular broccoli for that population. And then we can say, well, it's very interesting, the ones that have the long shelf life have this area of this linkage group, and then we can say that this is a marker for shelf life.
Maintaining the physical appearance of broccoli is one thing, ensuring it doesn't lose its essential nutrients so quickly is quite another. That's what researchers at Warwick HRI are turning their minds to now.
We've done some initial studies, and the main nutrient that we've looked at is vitamin C, and within about 3 days of being harvested, the lines that we've looked at have lost half their vitamin C content. What we've found so far is that the lines that do actually last longer appear to have higher levels of some nutrients, antioxidants, the things that the chemicals that actually are good for us in terms of anti-cancer agents and things like that, also seem to protect the broccoli head when it's been harvested. So it would appear that if we actually breed for longer lasting broccoli, it may be that we actually breed for higher levels of these nutrients.
So when can we expect to see this new longer lasting line of broccoli in the shops?
Well, Syngenta, who are the company that are interested in this, they're funding part of this project, already have a brassica broccoli breeding program that they're working on. And if we come up with genetic markers that are linked to good storage, then they can cross those quite rapidly into their breeding populations, into their elite lines that they then will use to develop new cultivars. So, I mean, if the experiments work, then probably within 5 years, or at least 10 years, quite rapidly, it would be available in the shops.